delighted to be here today as a representative of a team that's developed this Origins of Life laboratory in McMaster University. The PI for this effort was Michael Reinstetter, a biophysicist. I'm an astrophysicist. Ying-Fu Li is a biochemist, kind of a dream team in some way for an astrobiology setup. I want to emphasize then that the key question that we're interested in the origins of life, one of the big questions is the role of information. Where does it come from? How does it develop? Can it evolve? The RNA World hypothesis provides a basis for thinking about that, seems to be active in all young cells, and coming to the talk earlier that we saw today, the kind of environmental problems that you face in building up RNA polymers. As you can see in warm little pond environments, you've got UV that can destroy you, you've got seepage out of bottom of ponds, you have hydrolysis fighting you against this, you've got a polymerization occurring at some rate in some kind of a media, and this is a beautiful experimental question that kind of defies easy, simple, theoretical questions too. So this has been our motivation in building up a laboratory, which starts given that we're supposed to have molecules, et cetera, around, nucleotides in this case, lipids perhaps delivered from meteorites, and the question is, what happens next? The first step, how do we get polymers? So as is shown in your diagram here, we all know the importance of these condensation reactions, particularly driven home by the work of David Deamer and his collaborators. And in the situation that we're interested in, warm little ponds or environments like this where we can easily have such warm, wet, dry cycles, as well as thermal cycles that are driving the condensation reactions. You see here some nucleotides laying down on some layer, which can be complicated. There can be lipids, clays, salts, various kinds of things mixed in this messy prebiotic environment. We have heat and driving the thermal aspects of this. The wetting and drying allows you to form in one cycle a dimer. And, there's some ability of these molecules afterwards during the wet phase. During the next dry phase, dimer can meet dimer. If in appropriate conditions, you have a former, and by this process, the important role of surfaces here can actually help this process along. So this is the perfect kind of physical and chemical setup that motivates our laboratory. As you see here, this is designed by us and built by Angstrom Engineering, a top-level technology company near Kitchener, Ontario, near our university. That wonderful, fanciful, life-generating machine in the fog there is their artistic presentation of our apparatus. What you have on the left pictures is Renee, one of our students working in this. It's a computer-controlled situation. We've got a temperature control. We have control of the wet-dry cycles, very carefully built in. I'll show you in a moment. We have pressures just going to 1 bar. We can put different gases into this. We have a radiation field, which is, if I can get my light going here, the radiation field is 8 lamps, carefully designed LEDs going from infrared to UV in these cylinder heads here, nicely cooled, so I will show you the conditions here. So looking into our lab, into the simulator, we've got various kinds of radiation fields shown you here. Our intent is to do this experiment as you'd find the conditions on other planets being the TRAPPIST-1 system. Here are 2 spectra. The solar spectrum is an example, and the TRAPPIST-1 star. So we are now putting together the program that will program in these radiation fields. Everything can be oscillated against everything else. While the radiation is, while the water, while you have wetness, that can screen you from UV, so at that point the UV would be off. When you dry, UV becomes a problem for you, so the UV field turns on. So all of these can be cycled in any manner that you wish for the given type of environment that you want. I'll show you this movie here. So we're looking in the chamber. This is the beautiful follow-up of the talk this morning. We don't actually have a wet pond. We do this by dews that appear and disappear to 85% humidity. So if you look inside the chamber, you see the dew developing. On the right-hand side, you see the relative humidity dry up at the peak. The dew appears. There's a slight difference between the plate and the background. So there's a dew developing, disappears. This can be beautifully controlled by the expert design of our engineers. And so we can very nicely produce any kind of wet-dry cycle with thermal cycling that you want to program in. It's taken us a few months to find the optimal procedure for this, but before you have a machine that works extremely well. So the way we prepare our mini ponds, these are mini ponds that are put on silicon wafers, 1 centimeter squared. You pipette on whatever ingredients you think should be in your pond in whatever concentrations that you want. Here are shown 4 of them, shown here, pipetting on. The important things during wet-dry cycles, sorry about this, you see that in the top here, there's many layers of vesicles that layer one on top of the other during the course of many cycles. These layers, there can be up to hundreds of layers here, one on top of the other, and inside of each of these you trap some monomers, dimers, etc., that find one another mobile between these membranes. They find one another, they link up during the dry phase. When you wet, these things break up into little bags eventually, and eventually you can encapsulate things. So I will show you just some of our first results here. First of all, microscopy, just with pure lipids, the typical one used in all experiments with the DCMP, with just a mixture of, of here, AMP and UMP put in here. So this kind of a uniform field looking down in lipid. In early, we'd go, one cycle takes about an hour to do, so we can do, in 1 week, we can get good way through 70 cycles, make the cycle as long as you wish, and effectively a minute, a month, a year, that doesn't matter. It's just the number of cycles that matters. Here you see the growth of rather large structures, 100 micrometers shown in here, connected by these kind of channels. Intermediate in the number of cycles, you have these larger structures here which are starting to break up into the smaller vesicle type range structures shown in here. The walls get thicker, and into this I'll show you where the nucleotides are in a moment. In the final phase, we get now a morass, quite a complicated structures. These larger ones have broken up. You have these vesicle-like structures now inside about 50 microns or so, as David Deemer and others have found in their experiments. And if I show you now, we're going to use autofluorescence now. These nucleotides will glow. They're excited, and by light coming in here, and the images now that you see here are actually emission from the nucleotides, looking at their distribution. So I'll go from our first stage to the later stages, the last stages sequentially. So here we see this uniform kind of distribution. Early on, we've got these large empty regions that are formed. The green is with nucleotides. They're still kind of spread out, but they're finding their way, being concentrated in those walls. A little bit later, a few more cycles on, you see that these larger structures are breaking up. Most of the nucleotides are being concentrated now in the walls by the very process we've been talking about. A little bit further on, you now see this very broken up kind of structure, and peering into those, on this scale, you see that nucleotides are encapsulated very nicely. We have various physical assays techniques by driving at these analysis for this, including neutron and here X-ray diffraction experiments, so we can run on the samples to determine what's the structure of the layers and where are the monomers in these layers. These can be followed up by molecular dynamics simulations. So here's the basic geometry shown down here. The X-ray machine in the laboratory diffracting off this bilayer as an example. This is the kind of 2D diffraction pattern. You get the Bragg peaks, which you can then explore. here we've got the various distance scale between the molecules, between the lipids. There's the bilayer here. And by doing the analysis with some computer analysis behind you, here the distribution here in this diagram of the intensity as a function of Q here. This shows the typical scales. The typical scale in your bilayers might be 4.9 angstroms. Disorganized nucleotides have a separation of about 4.6 angstroms. That accounts for this peak. And the organized nucleotides, separated by about the distance of a phosphodiester bond, I should say, are in this growing peak right down here. Now, if you follow this with time, you find this graph here, which is a question of number of cycles. We've gone to about 70 in this experiment. By the way, all this work is still unpublished. Please, this is the first steps, but this is just new stuff we're showing you for the first time, first results out of our lab. But you see the growing organized nucleotide fraction reaching up to about 10% now after about 70 cycles in these organized units. This graph doesn't tell you yet that they are actual polymers, of course, but they're organized like polymers. To follow that up, one wants to do things like gel electrophoresis as an example, as David Deemer was just showing us. So here is René preparing the samples. We have centrifuge cleaning things up, getting ready to put the samples in gel electrophoresis columns, which are shown here. So this is, again, another part of our laboratory. Our laboratory, by the way, is fully equipped for all the physics, biophysics setups, the biochemical assays, the various instrumentation we've been showing you. And here, if I can just show you, is the gel electrophoresis experiment. Here are two ladders, 100 base pairs, just a standard ladder everybody uses. And again, these molecules are negatively charged. The lightest molecules move the furthest down in the gel. That's the way this technique works. And here is the result. So this is the ladder. I showed you 100 base pairs. These are spread out a little bit because of salt effects, we think. But you can see in this pretty well resolved. We've gotten already to about 50 MERS, perhaps. We will check that. But this has been, with the rapidity kind of things, I'm very interested in what David's results were in his own experiments, but these seem to be working just fine here. We'll, of course, want to do sequencing of this work in the next experiment, in the next part of the experiment. But for what this says now, given the reliability that David has pointed to for understanding and interpreting gel electrophoresis data, this is hopeful that we're actually demonstrating this. So I would just like to conclude that so far in these experiments, we've been able to demonstrate under conditions of warm little ponds where we've got thermal cycles taking up to 80 degrees and down, allowing bonds to form, and dry wet cycles which allow for this alternate mobility and nailing things down to allow the polymers to grow, that we're very effective in growing polymers that probably matter. These have been encapsulated within these vesicles. We don't know how much of this RNA polymerization may occur inside the vesicle yet. That's the next thing, the next part of the experiment, to try to determine that. Ultimately, of course, a huge step ahead, we do not know if any of this material is capable of transcribing itself. And that is the giant leap that, of course, we're all interested in, the step towards some kind of evolution. But I hope that you find this, we feel that this is not only a robust way of examining physics and chemistry in an early Earth environment, but we will soon be able to answer the question, are we alone? By doing this, say, in a TRAPPIST-1 or any other kind of world you might want to dial up. We will simply dial up a star radiation field and be able to run these under a variety of conditions. So with that, I'd like to thank you very much for your attention.
Thank you, Ralph. To first question here in the center.
Hi, Tommaso Faraccia from ESPCI Paris. I wanted to know if you did perhaps some low-angle X-ray scattering to see if there is any supramolecular structures that have a larger length scale than single stacking distance. And the
the answer to that is that there's a range of structures that you do find. There's quite a distribution that you saw, but still fairly peaked. And this has been followed up by molecular dynamics simulations. So we think we have a good feeling between the molecular dynamics and the data as to, what levels of structure we should be seeing.
Yeah, Matt, I was just meaning that at that Q you are sensible to the distance between the nuclear bases, but if you go at lower Q, you can maybe look at the distance between the different layers, or if there is other organization of the strand of oligomers that you have inside.
Yeah, we do probe the distance between layers. I showed you that number, I believe, up there, and the separation between the lipids. We get all those distances pretty well.
Okay. And second, which is the range of concentration you think you are exploring during dry Whiting cycles?
Yes, I think these were molar type setups in these experiments. So, and I forgot to mention, we have equal one-on-one of AMP and UMP in the mixtures that we had here, which are actually good for building larger polymers, as is known.
I got a question over here.
Yes, Ralph. I just want to comment that implicit in your talk is the use of base pairing monomers. The AMP and the UMP are, in fact, the base pairing of RNA. And I think you have found that if we try to make these monomers out of a single nucleotide, either oligo U's or oligo A's, it doesn't work nearly as well as having both present that would be able to form a duplex strand and not just a single strand. And we're getting strong evidence that that's the case from some atomic force microscopy that we're doing, where we can actually see these strands accumulating on a mica surface.
Oh, that's fascinating. Thanks for that remark.
One more question.
Hi, Milena Popovic, Blue Marble Space Institute of Science. Have you run denaturing gels?
No, it's taken. I'm not a biochemist myself. We rely on collaboration with Dr. Yingfu's laboratory to help us out with that. It's been quite a trick just to get, as you know, it's very tricky to get RNA to go through gels probably because the nature of the molecule, they tend to ball up, etc. So it's been quite a trick even to get that data that I showed you. I'm just not sure of the extent of the experiment done with other gels yet. I think this is the first one we've got working after some months of effort. So we're still in the baby steps here.
Please thank Ralph.